Okay, so welcome to today's coffee seminar, which is part of the coffee seminar series. Today we're going to be hearing about the curation of multi-source genomic and phenotypic data for global genomic prediction in tree crops. And we have the wonderful Shashi here talking to us. Shashi is a postdoc in the Center of Horticultural Science, and I've had the privilege of working with her for the last couple of years. If we can just change to the next slide. For those of you that don't know me, my name is Elizabeth Ross. I am a senior research fellow in the Center of Animal Science, and I collaborate a lot with the people in the Horticultural Science Center, which is how I knew Shashi. And I've been asked to facilitate this meeting, and it's a really great pleasure to go ahead and do that. Next slide, please. And of course, as you all know, at UQ, we do like to start with an acknowledgement of country, and the University of Queensland acknowledges the traditional owners and their custodianship of the lands on which we meet today. We pay our respects to their ancestors and descendants who continue cultural and spiritual connections to country and recognise their valuable contributions to Australian and global society. And I'd like to just extend that today to the traditional owners of the places where all of you listening are, both nationally within Australia, but also the traditional owners of wherever our collaborators and people that are signing in from international locations, and acknowledging really the contribution that those people all over the world have made to today's modern societies. Thank you. Next slide. Okay, so we're going to start off with a little bit of housekeeping. Today's seminar is scheduled from 12 to 1. After the seminar has completed, there will be an opportunity for questions and answers. Now, if you do have questions and answers, if you do have questions that you would like answers to, please do use the Q&A tab, not the chat tab. So we'll only be taking those questions out of the Q&A tab. And if we don't get to your question in the allotted time, I'm sure that Shashi can reach out to you directly afterwards. Next slide, please. Okay, so as I said, we have Shashi here talking to us, a postdoctoral research fellow. Shashi is a crop geneticist, a data scientist, and a plant breeder here at Coffee. Within the University of Queensland, and she applies innovative plant breeding approaches and uses large-scale genomic and phenotypic datasets to optimize breeding pipelines to enhance productivity in tree crops, such as apple, almond, citrus, and blueberry and other horticultural crops. Her research explores the integration of novel breeding technologies, genomic prediction, artificial intelligence, and functional genomics to enhance crop improvement. And her research outcomes have featured in 14 refereed publications, and she's passionate about training and mentoring the next generation of plant breeders. Next slide, please, Shashi. Okay, so at this point, I'm going to hand over to Shashi to share with you her great work, and I will see you all at the end to facilitate the questions and answers. Shashi, take it away.
Thank you, Liz, for your kind introduction. Thank you for joining with this talk. It's a pleasure to talk about curation of multi-source genotypic and phenotypic data for global genomic prediction in tree crops. First of all, I would like to acknowledge the country too. I acknowledge the traditional owners and their custodianship of the lands on which we are meeting today. I pay my respect to their ancestors and descendants, and I recognize their valuable contribution to Australian and global society. In this talk, I'm briefly going to talk about what is genomic prediction and how genomic prediction provide a platform to aggregate genotypic and phenotypic data And the issues I have encountered when aggregating this data set, and some fundamental to data curation and standardization of those data, and the workflow I have established for data aggregation, standardization, and storage. First of all, what is genomic prediction? Simply talking, it is a statistical framework, and it uses genome-wide markers to predict genetic merit in individual for traits. And it enables selection without prior knowledge of marker effect or marker trait association. So when combined with other tools such as GWAS, it provides some information to inform discovery of candidate loci and genetic architectures for traits in individuals. If we talk about genomic prediction models, here you can see linear mix model, and we use vector of phenotype and some fixed effect and random effect and residual errors. There are other models as well. When we are using genome prediction, we can use different traits such as polygenic traits that control several genes, and then categorical traits or case controls traits such as disease-related traits. This trait can be related to yield or quality, especially when we are working with tree crop species or fruit crops. We are talking about quality traits or physiological traits or phenological traits or some trait associated with multi-environmental performance. Although we can use whole bunch of traits, when the trait has high heritability, we can expect higher accuracy and ability in genomic prediction. To conduct genomic prediction, we need to have training population. Training population is the population we have genotyped and phenotyped. So we have both genotypic data and phenotypic data for this population. So we can make models using these data sets. Then these models will be used on another data set, which is known as predicting data set, which we have only phenotypic data, in order to find out breeding values. These are called genome estimated breeding values. So genotypes with high predicted breeding values, we can again use in our breeding populations. And these values we can again use in our training population in order to enhance the model. So when we have training population data from multiple locations, we can jointly use this genotypic and phenotypic data set to evaluate G by E, genotype by environmental interactions. So usually genomic prediction widely used in animal breeding because we can have large training population easily with animal breeding, and those breeding programs are centralized. But it's slowly moving into crop breeding as well, especially when we talk about perennial crops. They have large vegetative production cycle. So for some crops, it could be many years. For example, apple, it varies from 5 years to 8 years. Then they move into the reproductive cycle. So when we looked at the reproductive cycle, we clearly see two different types. Some plant species are in direct flowering. That means in order to produce flowering and fruits, they need undergo some chilling effects. So once they exposed to those chilling effects, birds happen, and then only we can get fruits. Almond and apple are crop species coming under indirect flowering. But some plant species don't need those kind of chilling requirements. They can produce flowering and fruit directly. Citrus is a direct flowering type. So when we looked at genomic prediction, perennial crops can benefit from genomic prediction. However, these crops have other challenges. Most of these plants, tree crops, are self-incompatible. That means in order to get the fruits, we need to have at least two genetically distinct individuals in an orchard. And those are huge plants and with very complex genetic architecture. So it's very difficult to replicate those trials, and they have long juvenile period. For example, as I told previously, apple, it is 5 to 8 years, almond, it is 3 to 5 years. And since these are tree crops, we are interested in many traits. And when we look at the phenotyping cost, it is very high, and traits are interact with each other and highly complex. If we want to use traditional breeding approaches for these crops, it is very challenging. Therefore, plant breeders are moving towards genome-informed breeding strategies. It provides a reduced phenotyping cost, and it shortens the breeding cycle across multiple generations. Moreover, we can select promising individuals at early stages because we are mostly based on statistical analysis and genotypic data. However, when we looked at crop breeding programs, they are highly fragmented. So in here, you can see the distribution of three tree crop breeding programs, apple, almond, and citrus, and they are all over the world. And they have very different objectives. And when they um, record data, they are very fragmented and then very diverse. If you want to use this data set in global genomic prediction, you have to face some challenges. However, we need multi-source dataset for reliable prediction. First of all, it captures wide range of genetic diversity, and it's coming from different environments, so it can make reliable prediction across even novel environment. And it reduces the data biasness. And also it provides platform to share data and collaboration and to enhance model performance. So where we can find those diverse data sources? We can find data from publication, collaboration, or data repositories such as GDR, Genome Database for Rosaceae. Here you can see we have collected data for three different plant species, apple, almond, and citrus. Almond is our main crop. So in this presentation, I am mainly going to talk about apple. Apple is the main data set. So for apple, we have collected 10 data sets so far from 7,000 individuals, and they are from 550K SNP markers, and they have been genotyped using SNP arrays 9K, 20K, 480K, GBS, and whole genome sequencing. And they are present in different data format. Some are in actual genotypic uh, types like AGTC, and some are in AB, BB format, and some are in VCF format. And they have different ploidy level as well. When we look at the almond data set, those individuals were genotypes using different platforms as well. And data format is very actual nucleotide format or AAAB format or VCF format. Citrus is exactly the same, but we have very small data set for citrus currently, and they are mainly genotyped using SNP arrays. When we look at highly diverse data set like this, one of the questions popping up in your mind is how we can use this data set. In order to use this data set together, um, looking at the common markers or exploit the replication of allele across dataset rather than using the whole genome clonal replication is practicable in order to integrate these genotypic datasets. Then we look at the phenotypic dataset. At present, for Apple, we are interested in 6 different traits. They are quality traits and yield traits. For quality traits, we are looking at fruit weight, sugar content, and fruit firmness, and acidity and pH. For yield, harvest date, and flowering date. And since they are from different breeding programs all over the world, they are from 12 different locations. They have used different methods to phenotype and record data. And so they have used very different trait descriptors as well. For example, for fruit weight, some use average fruit weight, some just fruit weight, some single fruit weight. And they are present in different formats, such as categorical format, numeric, and present as a percentage. And for fruit weight, some use actual weight in grams or kilograms. And some weight, it is small fruit or large fruit. When we look at almond data set, currently we are interested in 9 traits. Those are nut and kernel traits. And when we looked at the methods they have used, they are very diverse methods. And for the citrus, exactly the same. We are interested in 7 traits, and these are quality traits and yield traits. They have used different phenotyping methods and very different descriptors as well. So when we looked at those phenotypic and genotypic dataset, we have seen several issues. I'm going to talk about some of the issues we have came across with our Apple dataset. So Apple datasets have been genotyped using different SNP arrays, 9K, 480K, 20K, 50K, and GBS data. So when we looked at those datasets, they have some kind of replication, but different platform gave different names for these individual markers. So when we have replicated markers, there are some issues like noise and unwanted replication. So before connecting this data set, we have to correct them as well. And then we looked at the genotypes. In genotypes, previously we have found there are some synonyms. Synonyms are multiple names that are given to same clone. They have exactly the same identity. And homonyms. Homonyms are, we can see very different genetic identity or clones with same names. If our data set has synonyms or homonyms, there are going to be issues. So we have to correct them before integrating them into a one big data set. And phenotypic data. In phenotypic data, the most common issue we came across, the same trait measured with various phenotyping methods, and they have labeled differently. As I told previously, fruit weight is a classical example. And pH, sometimes pH they have measured using pH meter or they have used the titration method. And harvest date, some data set has actual harvest date or some has Julian dates. Julian dates are the dates from July 1st up to the harvest date. So if we are integrating this data set, we have to correct those errors to unify those differences. In that case, data curation is come into handy. What is data curation? It is the systematic process of collecting, cleaning, organizing, and integrating data. Once you curated data, you can have accurate, consistent, and standardized data set. Then we can combine those genotypic data and phenotypic data into a one big data set that can be used for downstream analysis. So how we can standardize our genotypic data set? So we have seen there are various names for individual markers. So instead of using individual marker names, if we use chromosomal position, it will be easy to unify this data set. To do that, we establish master marker file. So to establish master marker file, we find out all the sequences from Apple SNP arrays, and then we use those flanking sequence to align with the Apple reference genome, golden delicious. And we found out the chromosomal position for each of these SNPs. Then we aggregate those SNP position into a SNP database. Some of these SNPs are unique hit. That means they only have one exact position in the Apple genome. And some SNPs are multi-hits. They hit several positions in the Apple genome. Or some didn't say, came up with any significant hits. We label all those SNPs accordingly to standardize our data sets. So once we find out the exact marker positions, we use those data in order to find out unique and replicated clones. To do that, we looked at the marker profile and create the genome relationship matrix. Based on the genome relationship matrix, we identify unique and replicated markers. In a within data set, if we found exactly the same second sample, we label those sample as NTT. That means not true to type. So in the downstream analysis, we can identify those sort of individuals and deal accordingly. So now we find out the synonyms and consolidate into one record. And then we compare those with individuals in a foundation data set. We establish this foundation data set with the Apple clones that widely used by the Apple research community. In this data set, we have over 2,000 Apple clones. So we convert the clone ID into standard clone ID or CID standard to use in all our data sets. Simultaneously, we identify the homonyms and corrected them, adding the location name into it. As the last step of genotypic data curation, we looked at the individual marker profile and find out the ambiguous marker data. Because if any ambiguous marker data present in the data set, they could add further noise. So clarification of genetic entities with ambiguous markers are essential. To do that, we compare the genetic profile of individual with the foundation dataset. If we found any uncertainty marker calls, we recorded as missing. Now we have finished standardization of our genotypic dataset. So we use standard CID standard to standardize our phenotypic dataset. Within dataset, first we cleared our data. Especially we looked for mislabeling samples. And then we standardize trait descriptors. Especially we harmonize across the measurement unit because some are in kilograms and some are in grams and some are in ounce. So we harmonize those measurement into gram, and we uniform our data format as well, because in some data format it could be either year, month, date, or some in month, year, date, like that. So we uniform into one format, and we align all the scoring scale into one scale. And further, we looked at any differences in location code or experimental design. And across dataset, we ensure unique identities across accessions and genotype codes. And we further looked at any conflicting genotypes and inconsistent data or unreliable data point and make them missing to um, um, aggregate those data sets. So now we identify some of the issues present in those data set and the way to correct them. Since we have large number of data set, we established a structured semi-automated workflow in order to do all those curation. So it is simply talking, it is a three-step process. In step one, we take all the original data and reformat it into data contribution workbook. That is the format we have created to generate all those original data into same data format. When we are creating our data contribution workbook, we used our master marker file in order to find out the marker names and a genome identity to find out the CID alias present for clone names. Once we prepare our data contribution workbook, then we conduct all data curation. That means identifying synonyms and consolidating them, and identify homonyms and correcting them, remove polyploidy individuals, and then remove replicated markers, all conducted in step 2. Once we done that, we create the DCW curated version. And we use this DCW curated to create BIMS template for phenotypic data and GIGWA uploads for genotypic data. Because at the end, we are going to upload our phenotypic data into BIMS in GDR and VCF file into GIGWA. The BIMS template and GIGWA uploads are the main outputs we are obtaining from this workflow. For in this workflow, we establish in-house written R script, because using R script, it easily record reformatting and correcting any wrong entries or historical tracking. So what are DCW or data contribution workbooks? These DCWs or data contribution workbook are workbook with 12 templates that comprehensively record data about the data set. So for example, in here, marker BIMS template, we record all the information about the markers present in that particular data set. And CID BIMS, it's record all the information about the clones, including alias for that particular clone. And the other templates record all other information required for this particular data set. Once we prepare the DCW1, then we are ready for data curation. So we are preparing GRM and comparing it with the foundation data set. Once we have done all these things, we prepare VCF file to be uploaded into GIGWA, and BIMS template file phenotypic data to be uploaded into BIMS in GDR. For Apple dataset, when we conducted all these steps, we have identified 51 synonyms and we consolidated them into one record. And then we identified 7 homonyms and corrected them, adding location information. And we have identified 105 ambiguous marker profiles and considered as missing. Now, for genotypic data, we have curated SNP data. So we prepared VCF file together with the sample information and metadata to be uploaded into Gigva. Gigva is an open source and it is free data management system, and we are going to upload this genotypic data into a secure account. And once we ready to do genome prediction modeling, we can download data and use in prediction modeling. And then for phenotypic data, we are going to upload into a secure account in BIMS in GDR, or breeding management, breeding information management system. So these BIMs, they have their own template. So we can't use curated DCW directly. So we convert those curated DCW into BIMS format before uploading them into BIMS. So once we uploaded those phenotypic data into BIMS, we can download them easily to make prediction models. So we first obtain our raw messy data set and identify the issues with those data set and find out way to handle those issues. And now we have curated consistent data set. So we can use then in downstream analysis in breeding and genomic application. For Apple dataset, we have consolidated over 600,000 case SNP from 7,000 individuals. And we could identify some synonyms and homonyms, and we could identify some inconsistent markers as well. So we established some workflows. We have used those workflows in other dataset in Apple almond and then citrus. In further, once we do genomic prediction modeling, we can combine those dataset with GWAS to find out important variants, and we can do G by E by M analysis as well. And also we can use the GWAS results, especially large effect QTLs, can integrate with the GP models to improve genome prediction. And also this data set we have prepared is very big data set that captures wide range of genetic diversity. So we can use this data set in genome diversity studies as well. Before winding up, I would like to acknowledge all the contributors so far in this project. And I would like to acknowledge our funding body, USDA. And at last not least, I would like to thank you all for listening to my talk. Thank you. I'm happy to take any questions, and I'm hand over to Ross for chairing the question and answer session. Thank you.
Thank you so much, Shashi. That was really fantastic. Just to remind anyone, if you do have questions, pop them into the question and answer tab on the Zoom meeting. So we might kick off with a question from David. And his question is, can this process work with whole genome SNP data?
Yes. We have some whole genome sequence data as well. So currently we are mostly doing SNP array data. Then we use it for GBS as well. Our next step to fine tune our workflow for whole genome sequence data as well.
Excellent. Thanks, Shashi. Just a little note from Christine. She says, thank you for your talk, Shashi. If anyone else has questions, pop them in there. But in the meantime, I do have a quick question. And I was wondering, how and why did you select those specific traits for genomic prediction?
So these traits have moderate to high heritability. So genomic prediction works a little bit better for moderate to high heritability traits. That's why in this stage, we selected those traits.
Excellent, thanks. Just adding on a question from Christine. She says, how does the date record environment?
In we have some environmental data at the moment, but those breeding programs highly varied, and we don't have um, complete capture of environmental data yet.
Yeah. She's put in a clarification, but that is what you've asked. And she follows up to say, do you have the geolocations for the phenotypes?
Yeah, some data set we do have geolocations. Yeah, we are going to use that information in our model. Yeah, thank you.
Yeah. And another question from Leela. Hopefully I've pronounced your name correctly, Leela. They say, what is the minimum population required for developing genomic prediction models?
I think for plant or crop species, we need at least a couple of hundred individuals. So if it is less than that, your prediction model may not be accurate. Yeah. Mm.
And there is another person, I think a related question. Training populations should capture as much of the species genomic variability as possible. How big is big enough populations in tree crops?
Tree crop breeding programs, they vary. Some breeding programs are very small and some are a little bit larger. Small in the sense, some data set I have about 77 individuals, and big data set means 2000 individuals. I think integrating all those data set will make a better data set for genomic prediction. But if it is a smaller breeding program, it is very hard to do good genomic prediction. Yeah. Mm.
Excellent, thank you. Another question from, I'm going to attempt your name. I am sorry if I mispronounce it. Dinesha. Dinesha says, I was wondering whether you have both the SNP and RNA data for these crops or only SNP data.
I only have SNP data because at this point we are only going to look at the genomic data set, not RNA data sets. Yeah.
And another question from as Jad. Sorry about my terrible pronunciation. Can this model use the descriptive trait data such as fruit blush color?
The issue with the fruit blush color is heritability is very low compared to other traits. But if you have um, larger genotypic and phenotypic data set, this model can be easily trained for that as well.
Thank you. One more question at the moment. When integrating the phenotype data from multiple studies, do you need to apply a correction for unwanted batch effects?
It's a very good question. Yeah, we can use a standard deviation and then we can use corrected phenotype, and it will give better results out from the genomic prediction.
And Leela again asks, can we use segregating populations like F2 for genomic prediction, and what are the limitations on that?
This is tree crops. In tree crops, F2 population is highly heterogeneous and heterozygous. So for tree crops, we can use F2 populations. But if we are using annual crops such as wheat and barley, I think F2 genetic diversity is not that enough.
Thanks. And another question from Christine. Christine says, how are different management actions taken into account, and is that an issue?
Yeah, management action is an issue, and that's why we are trying to do G by E by M analysis as well. But at this point, for our analysis, we are not going to include management practice, and it will be included as a fixed effect.
Excellent, thanks. Well, there's no more questions in the chat at the moment, but I think that was a really good discussion, and thank you to everybody that did ask questions. I just wanted to thank, as we close off, just wanted to thank everybody for listening today. And if we flicked over to the next slide, I'm hoping that you'll all tune in next fortnight to hear from Professor Catherine Proper from the Northern Arizona University about her presentation From Cells to Ecosystems, Understanding Environmental Contamination. If you haven't already, please do sign up for the seminar invitation list, which we'll pop up the QR code in the next slide. Do sign up for that list so that you know what seminars are coming up and you can get nice reminders for them. So thank you again for everybody. Thank you, Shashi, for the time to share this work with us, and for all of the Coffee Seminar organizing committee and team for making this series so successful. Have a great day, everybody. Goodbye.
Thank you, everyone.